Hi, my name's Ted Pavlic, and today I'll be presenting the work of a former student of mine, Taeyeong Choi, who's currently a postdoc in the UK and wasn't able to attend to present this himself. Now, when we started this work, our motivation was thinking about the video analysis of social systems. And of course, for this audience, we were thinking of social insect systems. So if you have two systems that are otherwise very similar, but in slightly different contexts, then our question was, how could we determine what individual level behaviors were correlated to that higher level difference? And so, for example, if you have two isolates of a social insect colony, you might have one that has no queen and the other one has a queen, and you're interested in what are the worker level differences that correspond to whether the isolate is queen right or not. Or maybe there's a reproductive hierarchy more generally that is in place in one group but is being formed in another group. Or maybe one group is currently being alarmed while the other group is quiescent and not alarmed. And so we're looking for these individual level differences, sort of screening for individual level differences that we might then further investigate in later experiments. Now, the traditional way to do something like this would be to track all of the individuals or features of the individuals in those video frames. And so there's a lot of great work that's been done on this tracking, where you can either locate maybe a centroid of an ant and follow that around for every ant, or even down to articulations, joints, and follow all of those joints around and try to capture even smaller scale features of each individual. Now, the downside of all of this great work that's been out there is that it's still extremely costly to go through all of this whole process to generate data that then you have to take and then use another pipeline to analyze that data to even find those differences. So it's like a very costly pre-processing step. And on top of that, it introduces artifacts. So a stationary individual might have a track that moves around a little bit. And so that movement in that track, which is totally an artifact of the tracking, might be misinterpreted as a difference in the behavior of that particular individual. And on top of that, even choosing what you're going to track requires prior knowledge from human experts, which may or may not be relevant to the thing you actually care about. Now, we're much very more accustomed with using deep neural networks like this deep convolutional neural network here that when trained to do a particular task automatically identify important features from raw video data. So in this particular case, you've got a population of faces and you've got this multi-layer network, and at different points, if different layers in this multi-layer network, then it extracts different features at different coarsenesses. And so at earlier layers, it extracts patterns of local contrast. At later layers, it assembles those patterns of local contrast into patterns of facial features. At even later layers, it assembles those facial features into properties of faces, like this is a smiling face, this is a frowning face, and so on. And that ultimately allowed it to do a calculation like count the number of smiling faces in this cluster of faces. And so it automatically figured out the features that it needed. And so the thought was, well, if we show videos of, say, a colony of ants, and we train it to, say, for example, figure out the probability that a short video segment is currently in a stable reproductive hierarchy, then in order for it to do that task, it's automatically going to find features of interest from that raw video data. So it will find, again, patterns of local contrast. It'll assemble those into some sort of important patterns of features of social behaviors, which will then be assembled into the reproductive hierarchy status, which then can do the calculation that we trained it to do. And on top of that, there are now tools that allow us to then go into those layers and pick out from those layers and ask, what important features have you found that helps you do this prediction? So in this sort of example here from this paper on Grad-CAM, which is the tool that I'm talking about here, in a neural network that could identify elephants from scenes, you could then ask it, what are you looking at in the scene that tells you that you see an elephant? And it's able to put a heat map on the things that it identified that were the elephants. So then likewise, if we had this social group, if we ask it to predict this higher level thing, like reproductive status, then we can ask it, okay, so what things in the image allowed you to predict that reproductive status? And it can put a heat map on that. So the model system that we used to test this idea was Harpegnathos saltator out of Juergen Liebig's lab. And so these Jerdon's jumping ants are sort of the social equivalent of the famous experiment of watching the pitch drop, where you've got tar that is fed through a funnel and it forms these very slow droplets that if you then analyze in real time, you actually get a much better understanding of the process of droplet formation in general. And so in this case, you've got these ants that can go through a social transition, like going from being quiescent to being alarmed, but they can go through it over a very long period. So you can remove their reproductives at a particular time. It will take them about 20 days to form a new reproductive hierarchy. It's clear when the hierarchy is formed. It's clear when you remove the reproductives when the hierarchy was lost. It is not quite so clear the process that goes on in order to reform that hierarchy. However, there are certain behaviors that already have been known from just human researchers, human observations, that seem to occur during this transient period, like this behavior of dueling, which there are these two ants in this video that you've seen have been going through bouts of dueling while I've been talking here. So the sort of stereotyped back and forth behavior. So our thought was we can take video of these transitions over these 20-day periods, and we can take little, say, 20-second segments from different points along this timeline. So we can take segments like you see being iterated through here. It goes to black in between these segments from a couple days before we remove the gamma gates. We have a couple days after. You can see a bunch of dueling happening. We have days after that. You see additional dueling. And then close to when the hierarchy gets formed again, there's a lot less dueling and things kind of go back to normal again. So the thought was that if we could feed these into a neural network and it could find the dueling, then we would sort of, without knowing about dueling ahead of time, then we've kind of show that in principle this could work. And then on top of that, if it found other behaviors during that transient period that had not been described, then that would end up demonstrating that this process can actually help us discover new things as behavioral ecologists. So how do we do this in a little bit more detail? The first step is we pre-process those video segments, and we actually don't feed it the raw video exactly. We feed it optical flow information, horizontal and vertical optical flow information. So all it gets are these segments that kind of look like this fuzzy stuff here, which just indicates that there was movement from one frame to another. Those pre-processed video segments go into something called a deep support vector data description neural network. So this DSVDD is commonly used for anomaly detection, say for like cybersecurity. You can think of it as a type of principal component analysis or dimensionality reducing transformation, where it takes the high dimensional data from this cluster of like samples and it turns it into a cluster that has the tightest possible diameter, so it looks as much like a sphere in this reduced dimensional space. And by putting it into this spherical space, then that allows us to use the center of this sphere as a kind of simplistic model of this class of samples. And so if we're able to sort of compare samples during the transient period after they go through this transformation to where they are relative to the center of this point in this reduced dimensional space, the distance from there kind of becomes an indicator of stability. So the farther away, the more they're in the transient, the closer they're more closer to stable. Now that simplistic model isn't quite it doesn't capture all of the characteristics of what it means to be a stable Harpegnathos colony. So what we then do is we use what's called a generative adversarial network to generate fake ant trajectories that can trick our simplistic classifier. So we can actually generate fake motion patterns that are similar enough to real motion patterns that they can fake our initial classifier, but clearly, different because they were generated synthetically. And then we can build another classifier that can tell the difference between our fake trajectories and our real trajectories. And that classifier that can tell the difference between the fake trajectories and the real trajectories, when you feed it transient trajectories, can even better tell the difference between those transient trajectories, the real trajectories, and the fake trajectories. So in other words, it's able to kind of give you a stability score for any of these 20-second video segments. So when we test it out, we tested with a bunch of different benchmarks. So just the raw optical flow data, our DSVDD alone, a bunch of others that I won't talk about here. But basically, this inner outlier generator that we built does much better than all of the other benchmarks we picked here. So it does a good job at sorting things into the fake samples, the real samples from stable colonies, and colonies that are in this transient period just by this 20-second video segments. Then from there, we can actually go into our network and we can ask it, what are you looking at in order to be such a good classifier of stability? And so we use this thing called Grad-CAM, which allows us to sort of say how sensitive is your output to different portions of the input, and then paint kind of an importance map, which we can overlay on top of videos. And when we do that, what we find is that it finds dueling. So it's able to actually, in these are shots of dueling between these ants, and it's able to highlight portions of video where it found dueling. And on top of that, there are portions of video where there are ants right next to dueling ants that are doing things that might be superficially similar, you would think, to a computer to dueling, ants that are coming close to each other and so on, and it does not highlight them. So it really has to be the stereotyped behavior for it to end up highlighting. Now, there are other things that it highlights, and we haven't had the time to go through them and then see, has it found any additional behaviors that we haven't yet discovered that might indicate that there's other interesting processes that warrant further investigation. So that's an important next step. So in conclusion here, deep neural networks can automate the analysis of raw video data and then sometimes eliminate the need to do costly tracking at all. And this is not that different than already the kind of multidimensional scaling that people are doing with tools like PCAs. Those deep neural networks, you might have heard, are black boxes that are not to be trusted. But they don't really need to be black boxes because we have tools that can look inside them and make them human interpretable, similar to analyzing, for example, the loading of a PCA. So our goal is not to totally eliminate the behavioral ecologist. Our goal is to complement the behavioral ecologist, to create sort of a knowledge co-creation between the human and the AI to generate knowledge about social systems like social insects. And that's all I'll talk to you about today. For more information, I encourage you to take a look at this paper that was published this year on this process. It actually was the best paper at Swarm 2021 when we presented it there. This is, again, the machine learning work was primarily done by Taeyeong Choi, videos of Ben Pyenson in the Juergen Liebig lab, and I'm happy to take questions now if you'd like to send them to me. Otherwise, feel free to digest the paper, and I'm sure if Taeyeong, you've reached out to him, he'd be excited about that too. Thanks for your time.